Cellular respiration is the chemical process that releases the energy stored in organic molecules of food in form of ATP. In cellular respiration, glucose is completely oxidized or broken down, releasing CO2 as the final product. Oxygen acts as the final electron acceptor and combines with hydrogen to make water. A total of 34 to 38 ATP are released. Cellular respiration can be broken down into three major steps: glycolysis, Krebs cycle, and oxidative phosphorylation. This is a diagram that shows an overview of the process. Glycolysis, which takes place in the cytosol, breaks down glucose to pyruvate. Pyruvate then goes into the mitochondrion, and a derivative of it enters the Krebs cycle. Both glycolysis and Krebs cycle produces ATP through substrate-level phosphorylation. The energy in glucose is transferred to electrons carried in NADH and FADH2. These NADH and FADH2 then goes into electron transport chain and produces ATP through oxidative phosphorylation. Let's have a detailed look at glycolysis. In the first step, a phosphate from ATP is transferred to glucose to make glucose 6-phosphate. Then glucose 6-phosphate is transformed into fructose 6-phosphate. Another ATP is used to add a phosphate group to carbon 1 to make fructose 1,6-phosphate, which is then split into two 3-carbon molecules known as G3P. You will notice in the steps so far, 2 ATP molecules have been used. An inorganic phosphate is then added to G3P, and in the process, NAD+ turns into NADH. We now have an 1,3-diphosphoglyceric acid. Notice that this molecule has a phosphate on each end. A series of reactions take place in which the phosphate group is transferred to ADP to make ATP. At the end of glycolysis, 2 net ATP are produced, as well as 2 NADH. The end product is pyruvate. The pyruvate then goes from cytosol into the mitochondrion. Several enzymes catalyze the conversion of 3-carbon pyruvate to 2-carbon acetyl-CoA, yielding a molecule of NADH and 1 CO2. The acetyl-CoA enters the citric acid cycle.